Hello, let's discuss about Golgi apparatus. This is for class 11 students. Let's start. One minute. Yeah, I have written some points about Golgi apparatus. First, who discovered Golgi apparatus? Golgi apparatus was discovered by Camillo Golgi. Okay, the name of the scientist is Camillo Golgi. He is a biologist as well as a pathologist. Okay, biologist, logist as well as a pathologist. Okay, logist. In 1998, he discovered Golgi apparatus. Okay, these are flat, disc-shaped sacs attached with each other. A cell may contain even hundreds, okay, hundreds of these stacks. Okay. Golgi apparatus, like endoplasmic reticulum, these are also scattered in the cytoplasm, but they are not attached networks. These are sac-like structure connected with each other, or sometimes it is not connected with each other. They are piled up with each other like cascades. Okay, like this. It has cisternae. One is receiving side, which face towards the endoplasmic reticulum. Okay, and one is exit side, which face towards plasma membrane. Okay, it is the receiving side, and these are Golgi stacks or Golgi apparatus. Golgi apparatus. Okay, so and many cells contains even hundreds of Golgi apparatus. Next point I have written, Golgi apparatus serves as molecular warehouse. Yeah? It serves as the molecular warehouse and processing station for products manufactured by endoplasmic reticulum. In our last video, we came to know about endoplasmic reticulum synthesize proteins. Okay, also stores lipids. As there are two types of endoplasmic reticulum, that is smooth endoplasmic reticulum and rough endoplasmic reticulum, stored lipids. Okay. So these are the products of endoplasmic reticulum. What happens? They are carried by small vesicles. Okay, these are the vesicles, and proteins are carried. They entered or received by the receiving side of Golgi apparatus. Okay, what happens when these vesicles entered into these Golgi bodies? They modified their structure. That's why it is a processing structure for products of manufactured by endoplasmic reticulum, that is proteins and lipids, stored lipids. And after processing, these vesicles burned off or detached from the Golgi apparatus and entered into the plasma membrane. Okay, these are burned off. I hope it is clear now. Now, let's move to the next slide. Here, unlike endoplasmic reticulum sacs, the flattened Golgi sacs are not connected. Okay, I already told that the Golgi apparatus are stacked with each other or piled with each other. They are not connected with each other. Okay? So, the receiving side is called as C side, which is towards the endoplasmic reticulum or towards the end of endoplasmic reticulum, and another one is trans side. Trans side, it is the exit side, which faces towards the plasma membrane. Okay, one side of Golgi stacks serves as receiving dock for transport vesicles produced by endoplasmic reticulum. I already explained that in the last slide. Okay, yeah. This is the receiving side, and it receives the transport vesicles from ER. Okay, let's move to here. Yeah, one side of the Golgi tract serves as receiving dock for transport vesicles produced by endoplasmic reticulum. And the third point is here, a vesicle fuses with Golgi sacs. Okay. What happens? The vesicle fuses with Golgi sacs. This is vesicle. Adding its membrane and content at the receiving site. Content means the proteins and lipids. Okay. That's why the vesicle integrate into the Golgi sacs. Products of endoplasmic reticulums are modified as they progress through the stacks. As the vesicles entered into the Golgi vesicles, the products, that is proteins, got modified inside it and move towards the exit side. The exit side is called as sipping side. Okay? Here, sipping side of the Golgi functions as depot, dispatching its products in vesicles that ward off and travel to other sites. Exactly, the vesicles, endoplasmic reticulum and Golgi apparatus acts as an endomembrane system where the protein first formed inside the endoplasmic reticulum, then carried by the Golgi apparatus where it is modified, and then it delivered to the cell membrane, and this forms a endomembrane system. Here, this is the nucleus, or okay. These are the nuclear membrane, or okay. And this is the endoplasmic reticulum. These are the network. Okay. And what happens? Endoplasmic reticulum. 1 minute. Let me change the color here. And from the endoplasmic reticulum, small vesicles, small vesicles here, detached from the endoplasmic reticulum, which contains proteins or lipids. Okay. We take it proteins. Then the Golgi apparatus are present. Yeah. These are stacks. And this is the plasma membrane. Okay. Then the plasma membrane, and draw these vesicles enters into the Golgi sacs. This is Golgi apparatus. This is ER. I am writing it ER. That is endoplasmic reticulum. This is DNA. Nucleus, nuclear membrane, okay, this is nuclear membrane, and this vesicles enters and processed inside the Golgi apparatus, then it bursts off. Okay, and this is the plasma membrane, enters, and this is the exit side, and this one is receiving side. Okay, in this way the proteins from the endoplasmic reticulum processed inside the Golgi apparatus, then it depot or exit from the Golgi apparatus, enters into the plasma membrane. The whole system here the whole system from this endoplasmic reticulum to plasma membrane forms the endomembrane system. Endomembrane system. Okay, because this endoplasmic reticulum connected with the nuclear membrane, then it forms the vesicle which carry or carrier vehicle for the proteins and lipids. Then it enters into the Golgi apparatus with its C side or receiving side. Inside the Golgi apparatus, the proteins are processed. After processing, it depot or exit from the trans side and entered into the plasma membrane. This vesicle, endoplasmic reticulum, and Golgi apparatus forms the endomembrane system. Okay. Okay. Let's summarize it. What I have told you, I'm just summarizing it so that it will be more clear. Okay. Yeah. We came to know that one side, the Golgi apparatus, has two sides. Okay, two sides. One is cis side or receiving side. What happens in this side? It receives the products of the endoplasmic reticulum. Okay, yeah. And second one is trans side or exit side. This depot or expel the processed proteins to the plasma membrane. Okay. Plasma membrane. Okay. Yeah. 1 second. This is the Golgi apparatus. Golgi apparatus are stacked or piled upon each other. They are not connected with each other. Yeah. This one is Golgi apparatus. Yeah. And these vesicles are bud up. Okay. This is the plasma membrane. Not only one vesicle, there's many vesicles at bud up from Golgi apparatus. Okay. Here, products, and it comes from endoplasmic reticulum. This one is endoplasmic reticulum. Okay. The first step. This is the first step, and this is second. They're processed inside each step. This is the third step, and fourth one, the bud of, this is 4. Okay. And this was 5th. This enters here. This side is the cis side or receiving side. And this one is trans side or exit side. This is PM or plasma membrane. Okay. This one is endoplasmic reticulum. I am writing it ER. ER for endoplasmic reticulum. These tracks are Golgi apparatus. Okay. Golgi apparatus. And the whole process here is part of or form the endomembrane system. Membrane system. I hope you got it. Okay. Okay. What happened? The vesicles are bud off. That is detached. Process I am writing here. The Golgi apparatus receiving. Receiving. Processing. And sipping. Products. Okay. This is all about the Golgi apparatus system. I hope it is clear now. Thank you. And it is the image I have uploaded from Google. Yeah, Golgi apparatus. This is the C space where it receives incoming transport vesicles from ear. Then this enters into the lumen of the Golgi apparatus, and this outer side is cisternae and inner side is lumen. Okay? Yeah. And finally what happens? It comes here. Finally what happens? This enters, and here the vesicle buds are, yeah, secretory vesicles, and this one is transverse or exit side of the Golgi apparatus. This is more clear picture. Here the DNA or mRNA products, that is mRNA ribosome which are present in the endoplasmic reticulum forms the protein. You can see it, ribosome. This is rough endoplasmic reticulum as it has ribosomes, number of ribosomes. Proteins are formed, and these proteins are carried by vesicles. Vesicles are the carrier vehicles, transition vesicles. Then vesicle bond off from the endoplasmic reticulum. Then it enters into the Golgi apparatus. Yeah. This one is Golgi apparatus. Okay. Then this proteins are processed inside this Golgi apparatus. After some time, it also burst off through the vesicles, and this vesicles enters this processed protein into the plasma membrane. You can see it. This is plasma membrane. Okay. And this is the whole functions of Golgi apparatus, and the whole thing here, from here to plasma membrane, endomembrane system. I am writing it EM. I hope it is all clear now. Thank you for watching.